Actually, this is the SAS Institute prescribed syllabus for the certification purpose. Okay, I hope you can
Okay.
see my screen here. So I'm just muting your mic. If you have any doubts, you just ask me. Yeah. So this is the syllabus given by the SAS Institute for the certification purpose. So this is the syllabus. Let us see clinical trial process. So the first part is about the theory only. Describe the clinical research process, means what are the different phases of the clinical trial. Okay, then what are the key roles, means key roles of different organizations and key roles of the different people who work in the industry. Then interpret a statistical analysis plan. We call it usually it as a SAP, statistical analysis plan. It is a hurt like for a SAS programmer, because every day we'll be doing the programming, or else we'll be generating the tables, listings and figures, okay, using the statistical analysis plan only. So the statistical analysis plan is very important for us, and every time we'll be reviewing the SAP. So when you get a job also, initially they will ask you to go, through some of the statistical analysis plan for a better understanding, okay, of the study. So interpreting the results, and whether your study is meeting the primary objective and secondary objective of the, the trial. So everything will be decided based on this SAP only, okay. And how the SAP is created, the SAP will be created by the biostatistician. So the biostatistician, based on the protocol, there is a separate team called the SAP team, Statistical Analysis Plan Creation Team. So the team usually contains the biostatisticians only. So they will create the SAP. Okay, derived from the protocol, because the protocol is the first copy which comes into the the clinical research process. So based on the, what we said, the protocol only, the data management plan will be developed, statistical analysis plan will be developed, the recruitment of the investigators will be done, and it contains the study design, how to conduct the clinical trial, and what happens, in which way you have to recruit the patients, and how to recruit the, the institutional review board, and how to treat the patients, and what are the medications given to the patients in case of adverse events. Okay, so everything will be there in the protocol. Okay, so anyway, the SAP also, it is a statistical analysis plan derived from the protocol, so that we will be discussing. Then deriving the programming requirements from an SAP and an annotated case report form. So this is also very important for us. Means, as I told you, we'll be always handling that SAP. Okay, so you should understand what are the programming requirements from an SAP. If somebody gives you the SAP, then you cannot simply use that SAP. First of all, you have to derive, or you have to make some rough work, like which process or which procedure or which type of programming is required to meet this SAP. Or SAP contains some, table creation or some mock table. So to create a mock table, then how you are planning, in which way you have to write the code, which variables we need, okay, what type of annotated case report forms we need. So these type of things will be deriving, okay, to create the content which is present in the SAP. Okay, later describe regulatory requirements also. So there are some regular requirements, because anyway we are working in the clinical industry, so that it's better if you know the clinical language very well. So what is happening behind, not only the SAS programming part, we should learn the ICH GCP guidelines also, and 21 CFR part 11. Then what is it, good clinical practice, and we have some theoretical concepts also, because you should have some domain related knowledge, the domain knowledge, okay, to work efficiently. So we are like sciences background or some pharmacy backgrounds people, so that we can understand the terminology very easily. Okay, see, for example, you know about the burns, like first degree burns, second degree burns, third degree burns, like that. So whenever you are working on the data, for example, if somebody forget to add that burns, simply write the degrees burns, then immediately you will find out the error. There should, there is no such word called simply the degrees burn. Something is missing, either first degree or second degree or third degree, so the digit is missing. So you can understand, okay, why it is missing. So it won't give any errors, but some logical errors you can pick it easily. Okay, so that is why usually they prefer the life sciences or pharmacy background people to work as a SAS programmer in the pharma industry. Okay, so second part is the clinical trial data structure. Data structure is nothing but the data set, right? So how the data is generated in the clinical usually after conducting the clinical trial or while conducting the clinical trial on the patients. What's the clinic? Clinic is nothing but patients, right? So there are two phases. One is the preclinical, the second one is the clinical. Preclinical means we will be testing the medicine or drug on the animals and in the laboratory. So that's called the in vitro and in vivo. So those are different. Then after proving your your drug as a safe and effective, then we will be conducting the clinical trials. Clinical trials means conducting the study on the human beings. So there are two phases in the human beings. One is NDA, IND, NDA. IND means investigation in new drug. Second one is the NDA. NDA means new drug application. What's an IND? IND means you are investigating a new drug. Okay, IND, investigation in new drug, IND. So in this IND there are again three phases: phase 1, phase 2, phase 3. In the phase 1, it is just the safety purpose only. In the phase 1 we will be recruiting 10 to 5 people or 20 people, not more than that. Only tens, not more than that, not in hundreds. It is just 2-digit number only. So let's assume we recruited 10 people, that to healthy human beings, those who are not suffering from any disease. Then apply your medicine on them. So here we will be doing some drug escalation studies also. What do you mean by drug escalation studies? Means how much the optimum dose. You have to decide the dose. Okay. See, for example, paracetamol, a man can survive 650 milligrams. Okay. Then if you take some alprazolam, that is a sedative, or some, then 5 milligrams or 0.5 milligrams is enough. If you give 650 milligrams of that medicine, then the patient will die. Okay, excess dose. So why because the doses will be measured in milligrams, micrograms, or grams. Okay, so how much the man tolerate. So these type of drug escalation studies, the minimum dose, minimum lethal dose, optimum dose, all these will be decided in the first phase itself. Okay, so once the drug is proven safe in the first phase, okay, then they will go to the second phase. In the second phase, they recruit the patients. Why? In the second phase, they have to prove the safety of the, effectiveness of the drug. Already the safety has been proven in the first phase. Then the second phase, they recruit the patient, then they will give the medicine to the patients and they will check for the effectiveness of the drug, whether it is really working on the patient, can it cure the disease or not. So, after second phase usually 200 or 300, just hundreds only, not thousands. Then if the results are proven safe and effective, safe in the first phase, effective in the second phase, then they will do the third phase. In the third phase, they recruit some thousands of people, maybe 2,000 or 3,000 or even 5,000 also, based on there is some sample size determination. So what is the sample size determination? It will be done by the biostatistician only. Anyway, that will be there in the protocol. Okay. So there is a separate material for the study population. How to make the population size. Okay, sample size. So that depends on the frequency also. See, for example, a peculiar disease is coming. That disease you can find one person is suffering in 1 lakh. Okay, or one person is suffering from maybe 10 lakhs. Out of 10 lakhs, one person is suffering from disease. Then you are conducting a clinical trial on that particular disease. Now your target is you have to conduct this clinical trial on 3,000 people. So out of 2 lakhs, one person is suffering from disease. Then if you are planning to conduct the 3,000 people, okay, 3,000 people, then how many patients you have to screen? Okay, 1%, 2 lakhs, 1%. Then 3,000 people means how many people you have to scan, and you have to screen. So it will be very difficult, right? It is very difficult to find out that much of the. Sometimes it may take years also to recruit only the patients. Okay, so it depends on the disease frequency. For example, there's a group, 100 people, 50 people are suffering from the same disease. Out of 100, 50 are suffering. Then if you your sample size should be no need of that much, because out of 100, 50 are suffering from the disease. Then take the sample of 50 only. Okay, 50 means just screen them and get, because the here the screening time will get reduced. So anyway, no need to bother about this one, because there is a sample size determination sample size determination techniques are there. So those techniques will be used by the biostatistician to determine the sample size. because when you read any trial, so the trial will show 157 patients are participated or 230 patients are participated, 33 patients. Why they are giving 230, 130, 137, 157 means it depends on the sample rate determination by the biostatistician, right? Okay. So after deciding the sample size, okay, then they will conduct the clinical trial, right? Then while the clinical trials are going on, then they collect the information into the case report forms. We were talking about the second phase, right? What happens in the second phase? Second phase is completely efficacy. Then how the efficacy is decided means they will check the adverse events. Okay, I mean, how the safety is determined in the first phase. First phase, safety is determined by the adverse events only. If patient has died, or if the patient is having some serious in any adverse event, then we can say that the drug is not safe. Okay, always the safety evaluation on the patient can be done in the first phase. Then second phase is effectiveness. Effectiveness means the patient was taking the medicine for 1 year but no use. So we can say that drug is not effective. It is safe. If you take the sugar pill, that is safe. But sugar pill, it won't cure any disease, right? In the same way, your medicine, it won't work. That's the second phase. Then coming to the third phase, in the third phase, you will determine everything, like you will consider everything, safety and evaluation. Then the third phase, we will be recruiting some thousands of people, maybe 2,000 or 3,000, recruit the 2 to 3,000 people, and then conduct the clinical trial according to the protocol. So the largest phase is the third phase only. Then after the third phase, then we will be generating a ISC report. ISS means integrated summary of safety, integrated summary of efficacy. Means first phase, second phase, third phase contains the safety, efficacy, and safety efficacy in the third phase. So from all the three phases, we will be generating one report. That's called the integrated summary of safety and efficacy. So there's a difference between the integrated summary of efficacy and end of study. What's the end of study? End of study means end of that particular trial, phase 1, end of phase 1 trial, end of phase 2 trial, end of phase 3 trial. So end of trial means you will be submitting only the first phase reports and second phase reports and the third phase reports. But when you are submitting the ISC means it includes all the three phases, three phases report as a integrated summary of safety and efficacy report. Okay. So then after completing this third phase, then send the reports to the FDA. Then the FDA, they will review everything, and finally they will give the approval to market your drug. Okay, then the drug will come into the market, then that will get distributed. But sometimes, 1 in 1 lakh patient may die. Okay, 1 in 1 lakh a person may die because of this drug. But you are conducting the clinical trial only on the 3,000 people. So in 3,000 people, a death may not happen, but in 1 lakh people, a death may happen. So you are not conducting a huge population. You are conducting just only few population as a sample. But when it comes into the market, it may cause maybe 10 deaths in a 1 lakh people. It may do. So the drug is not considered safe. So what we do, once in 3 months or in 6 months, again we'll be sending the reports to the FDA. Those who are using the medicine collect the information from the huge patients. Okay, but anyway, you just collect randomly. Okay, those are not very much strict. Okay, then later send the reports to the FDA. Maybe some 5 or 6 years. Okay, if anything reported, then if one death is happening in 1 lakh or some one death is happening in 10,000 people, then they will do the analysis and finally they will decide whether to retain the drug in the market or whether they have to ban the drug. So that is how so many drugs are banned in the market. Okay, I will show an example. Nice tablet, Nimsulide. Nimsulide. Earlier it was proven as very effective. It's a best anti-inflammatory drug. Okay, if somebody is suffering from pain or some, some body aches, then if you take a nice tablet, then immediately within half an hour everything will be cured. But nowadays the nice got banned in so many countries because of the adverse events. The same thing. 1 in 1 lakh people, so one death is happening. So that's the reason so many drugs are terminated or banned in the countries. Okay, so this is the this is about the phases, like first phase, second phase, third phase, and the fourth phase. Fourth phase is called the post marketing. So NDA means new drug application, means we are applying the drug into the market. We are bringing the drug into the market, and later we will send the report. That's called the post marketing surveillance. That's the fourth phase. Okay, so anyway, we'll discuss that drug development process. Okay, then approval process means first you create one new molecular entity. What is a new molecular entity? Means that maybe it is an antibiotic, maybe it is antiviral, or maybe it is a disease curing or anti-cancer, whatever it may be. You are developing a new drug. Okay, that what is the drug? It's a new molecular entity. Okay, anyway, the drug contains a molecular structure, right? So to develop a drug also nowadays, I think there are so many tools came into the market, like what we say, drug designing tools. Okay, then molecular modeling, like that we got so many tools in the market. So based on those softwares we will be designing the drugs, and finally the drug will be synthesized in the laboratory, and then it has been tested. Okay, so drug development process means how you are developing the drug and how you are conducting the clinical trial. So it all comes in the drug development process. Drug approval process means whenever you create a new molecular entity, then apply to the FDA. What is an FDA? FDA is a regulatory authority. Okay, so the regulatory authority will give the permission to market the drug, and to conduct the clinical trials also you have to get the permission from the FDA, US FDA, that is United States Food and Drug Administration. So every country is having their own regulatory authority. When you see the India, in this India there is a CDSCO, means Central Drug Standards Control Organization. So when you are planning to conduct the clinical trial or market one drug in the India, then you have to get the permission from the CDSCO. Okay, but why people are applying to the FDA? Because the FDA is it's the ultimate one. It's the extreme one, right? So when you apply or when you get the permission from the FDA, then getting the Indian CDSCO permission is not at all difficult. Got it? Because you passed in the uh, I mean some world level, then local is fine, right? Local authorities will give the permission. Okay, so it's not difficult, because the USFDA they have a standard regulatory authorities. Because you can see there are two types of, we classify the countries into two types: drug developing countries and non-drug developing countries. Okay, but most of the countries are not developing the drugs. Okay, only few countries are developing the drugs, but the majority of the population are present in these countries only. Okay, when you take the Africa, no, like Africa, it's not, it won't develop any type of the drugs. Okay, it will just import the drugs from other countries only. Okay, so so anyway, this theoretical content we will be discussing later. So what I am planning to explain is clinical data structures, means how to create the data sets. So how the data is generated or originated usually. While conducting the clinical trial, the doctors or the investigators, they will collect the data from the patients in the form of case report form. Okay, how they will collect the data? There are different methods. One is by observing, physical examination. Okay, eyes, nose, ears are good or not. How is the skin? Okay, they will do. That type of the data collection just by observing. The second one is by questionnaire. What is your age? Okay, then are you married? Are you educated? So based on the questionnaires, the data will be collected. Then based on some screening means they will check the blood pressure. Okay, they will check the hemoglobin. Like this, some screening, pulse rate. Okay, then heart rate. So this is the screening things. Later they will collect the information from the laboratory also. After perform some laboratory tests, blood chemistries, then the data will be generated from the lab. Means there are different ways to get the data from the patients. Questionnaires, by observing. Okay, by physical examination, that is by observing, and by laboratory test, and by taking some measurements. So there are different ways to collect the data, and all the collected data will be consolidated together. Okay, and we'll build a database. Okay, what is the database? The database contains the complete history of the patients who participated in the clinical trial. Then once the data is collected, is it possible to send the data as it is to the FDA? No, the data will is not arranged in a proper way. Okay, so what we do, we break the data into different domains. Okay, so how to break the data into different domains, and what are the standard domains? So these type of clinical data management things will be there in the CDISC website. CDISC means it's a clinical data interchange standards consortium. Okay, so it contains all about the data standards only. The first thing is how to conduct the clinical trial. There's no any like authority, the regulatory authority instructions about how to handle the data. But the second part is about how to handle the data. It's not like how to conduct the clinical trial. It's all about how to handle the clinical data originated from the clinical trial. So, I mean, after getting the data, we'll be breaking the data into different domains like this. Demographic data separately, lab data separately, vital signs separately, then adverse events, concomitant medication, physical examination, ECG, radiography. Like this, we are splitting the data into different domains. Okay, so those are called the SDTM data sets. We'll converting them into the SDTM data sets. Later, after converting the SDTM data sets, then we combine some tables to create one report. For example, you are creating a report which contains what the adverse event experienced by the patient and what are the medication given to the patient for those adverse events. So this type of table I want to create. Okay, I want to create a report. What's the report? What are the adverse event experienced with the patient and what are the medications given to the patient to save from that adverse event. So this is the report we have to create. Then where to get the data? Adverse event data set we have separately, and the concomitant medication given to the patient is separate one. Then you have to merge both the data sets into one common data set, and that data set will be used to create the report. Okay, so this is called the derived data set. What's a derived data set? Means we have multiple data sets. So whatever the variables required for us to generate one data table, then we are deriving the required variables from multiple data sets and we are creating one different data set. That's called the analysis data set. What the analysis data set means? We use this analysis data set for generating the report. Okay, so first collect the data and split them into SDTM datasets. Later combine the SDTM datasets to convert into the analysis datasets, and from those analysis datasets we will be creating the reports. Okay, our tables, listings, or figures. We call it the TLFs or TLGs, and send this to the FDA. So this is the process. So, but what's the defined.xml? Defined.xml means how you are doing the SDTM and ADAM, okay, how you are arranging the SDTM ADAM. So all these will be defined in a form of a one XML file. So that's called the defined.xml. Okay, so this is the, anyway we'll be learning and doing practically all this. Then coming to the SAS programming, how to import and export clinical trial data. Just now we discussed the data will be collected into the case report form. Then from the case report form, how to annotate and how to convert into the databases, and what type of case report forms are there, like electronic case report forms, electronic data capture, paper case report form, then how to collect the data from the case report forms, then how to consolidate. If the data is collected in the Excel, how to import, or if it is collected in a Oracle database, because you know the Oracle Clinical. So Oracle Clinical is a very sophisticated clinical trial software. Most of the companies are using the Oracle Clinical, and some companies are there. They are using some open sources like Open Clinica. Okay, then some use VBA. Backend will be the Excel. Like this, different companies, they use the different databases, and how to import the data from those databases and convert into your SDTM datasets. So that we will discuss. Then how to export and how to transport it, okay, to the FDA. Then how to manage the clinical trial. So here we'll be discussing about the dictionary tables. You know the dictionary tables, right? Dictionary table means when you open the SAS environment here, you can find a lot of things in the SAS help library. Okay, there are some catalogs. So when you open this SAS help, you can see there are some catalogs. In the catalog, we have different components, right? These are the graphs, and these are the reports. So what these are? Okay, these are called the dictionary tables. Means the information about the current session and the content present in the libraries. So it's called the dictionary tables. Anyway, I will explain the dictionary tables also. Then examine and explore clinical trial input data. So this is very important one. Means it is how to manage the clinical data, but usually the statistician, they will decide this about finding the outliers, missing, zero values, but I will be explaining with 1 or 2 examples. Then this is the common clinical programming, transforming the clinical data. So here we will be learning the clinical programming techniques, how to categorize, what's the windowing, what's the imputation method, last observation carried forward, then baseline observation carried forward, then worst observation carried forward, how to transpose, what is the change from baseline, then how to count the events, then Actually here they have given some 5-6 points only, but we will be learning more than this. Okay. Then how to apply the statistical procedure. It's very important. I don't know whether you are familiar with the statistics or not, but you know SAS means the SAS means itself is saying that it's a statistical analysis system. So every person, I mean, who is acting as a SAS programmer, they should have some statistical knowledge. Okay. Here we'll be getting a SAP. Okay, then based on the SAP we are doing the programming and submitting the reports. So that's fine. But instead of doing blindly, if you know what is what, see for example, if you know what is the univariate, where it is used, what is the frequency and where it is used, and what type of frequency better to use to get the reports. Okay, so if you know some little bit of statistical knowledge, at least if you can interpret the result, then that will be fine. Okay, so here I will be explaining you some statistical process from the basic. Maybe 3-4 hours I will spend on this statistical part. Okay, so what's the p-value, what's the one sample, what's the paid and two sample t-test, univariate, mean, summary, what's the n into p and 2 into 2. So because the SAP is the heart, I told you, right, statistical analysis plan, so that contain what statistical process you have to use. So we'll be discussing the statistical, I mean maybe 2 to 3 hours. Then coming to the macro programming. So macro programming means, you know, I think you might have learned the macros in the advanced, but here will be just, it is just like a brush up, because complete coding, the SDTM coding, ADAM coding and everything, define XML, everything we use the macros only. So without macros it's very difficult to write the code, because the code will become very longer and we will be writing the code again and again, the repetition things will happen. So to avoid those repetitions and making your program perfect and creating multiple reports using a single code, and for automation purpose also we'll be using the macros only. So anyway we'll discuss the macros. Then coming to the clinical trial report. So PROC report is a, you know, that it's an ultimate procedure to generate the reports. Every report or every table or listing, whatever we generate and output in the clinical trial, so that will be done by the PROC report only. So you should have an excellent command in the PROC report. Okay, so I'll be explaining the PROC report also. Then coming to the validating the clinical trial. So there is a separate topic called the validation. So this is very important one. What's the QA and QC? Quality control and quality assurance. Okay, so quality control means, see for example, okay, I think you might be knowing this, but simply I'm explaining. You're purchasing a car, right? So when you purchase the car, they will give the quality assurance. Okay, quality control means, see, if you want to prepare a car, it having 4 wheels, then a body and the color, so multiple things are there, right? So a person who is like doing the wheel alignment or who is fixing the wheels, okay, his work is only to just fix the wheels. So he will check the QC, quality control means when it is related to one area, it's called the quality control. So for example, 10 people they prepared the car, so 10 people each one they will do the QC, and finally before delivering the car, then you will test the overall performance of the car. If the overall performance of the car is good, then we call it as a QA is okay, quality assured, means we are giving the quality of all the all the parts of the car. So this, overall is called the quality assurance. Then quality control means it is related to the particular. Okay, so here we will be learning the QA, means when you are submitting the report to the FDA, overall quality you are checking. Okay, not only in the data, okay, in the reports, in the tables, in the coding, in the SDTM, and in the ADAM, whether it's meeting the SDTM in the FDA standards or not. So you'll be checking in all the areas. Okay, then coming to the validation. Okay, here we are talking about the data validation. Okay, data validation. So data validation means we have different programming techniques to validate the data. Okay, usually we call it as a data cleaning or data validation, edit checking or error checking, debugging. So we use different terms for the validation of the data. So I will show with some errors. Error handling methods. So, there's so many errors, right? Syntax error, logical error, data error, semantic errors, execution errors. So we'll find different types of the errors. Okay, so there are 2 books especially on this validation, and some tools also there in the market for data cleaning purpose. There are separate tools are there only especially for the clinical data cleaning. Okay, So this is the syllabus prescribed with the SAS Institute, right? But this is not enough actually. Okay, we should understand the final study report, how it looks like. Then it contains no any TLF creation, right? So we should know the TLF creation also, what type of tables, figures we have to create. Then there is one MEDRA coding also there. Okay, medical decision for regulatory activities. So how to use the MEDRA coding. So we have to discuss a lot of things behind this. Okay, so yeah, this is the simple overview of the syllabus, and also yeah, so this is the just little bit expanded one. Introduction to the drug development and discovery process. Overview of the highly regulated drug development process from discovery to bringing a biopharmaceutical product to the market. Understanding major phases, 1 to 4 phases of clinical trial and data management. Learning rationale for each phase of drug development cycle and introducing role of SAS programmer in each phase. Food and Drug Administration regulation and guidance, 21 CFR Part 11 GCP, good clinical practice and electronic common technical document, which will be sent to the FDA after completion of the trial. Then regulatory overview and approval process, including IND, NDA to the FDA, investigation new drug and new drug application. Then role of SAS programmer at any pharmaceutical industry. Then types of parameters of clinical trials. Then getting in-depth knowledge about the functional group and working of CRO pharmaceutical industry. Okay, and knowing in and out of guidelines pertaining to company involved in the clinical trials. Learning about the general department structure of pharmaceutical industry, roles and responsibilities of the SAS program in the company. Then getting to know what standard operating procedures and guidelines are in, and its importance. Discussing and reviewing the standard operating procedures and guidelines from a real pharmaceutical industry. Understanding the clinical study and documents like protocols. As I told you, the protocol, you should understand the protocol, at least one sample protocol. Then case report forms, another case report form, electronic case report form, annotations, then statistical analysis plan, etc. Get a feel of folder structure adopted by most of the pharmaceutical and CRO companies. So you should know this one. Get a feel of folder structure means how organized. How organized the data and everything. So you should get a complete concept of what's the work of the programmer. Okay. Then learning more about types of analysis in the clinical trial. Pharmacokinetics, pharmacodynamics, safety and efficacy. This type of analysis we will be learning. Then introduction to the clinical trial data. So it is just the theory introduction. Then this is the working clinical data preparation issues. Learn to prepare and clean the data. Know how to categorize and summarize the data. Studying and classifying different types of clinical trial data, ISS, ISC. Then getting acquainted with new clinical. These are CDISC standards. Okay. Then working with real pharmaceutical industry data like demographic tables, concomitant medication. So these are all the domains in the SDTM. Then this is a data analysis data set creation. It's ADAM. Then while creating the ADAM, we use the meta dictionary, WHO, time to event, change from baseline calculation. So it contains the SAS programming on the clinical data, then generating the reports, tables, listings and figures. Okay. So this is the complete syllabus. Okay, 4 parts. This is one I'm going to explain, but anyway, usually what we do, the programmers who learned the base and advanced from some other trainers from SAS Institute, okay, they might be having the knowledge of the statistics, right? So that's the reason I am not going to explain the statistics for them. But here, anyway, I will explain the statistical part also. We'll follow both, okay, my syllabus as well as this SAS Institute syllabus. Okay, I will be explaining both the contents. Okay. Now, yeah, my request is I'll be sending you one folder to you. Okay, I will just upload one folder. Every day you just download the content from that folder only. Okay, the folder contains some sample videos. Now I will be uploading like 3 or 4 hours videos. Okay, that because I can't explain the complete clinical research process. Okay, because already you have some basic knowledge of clinical research and drug development, right? So I'll be uploading 3 or 4 hours of the the classes which are related to the clinical trials. Okay, that is completely theory only. One is about the SAP, about the protocol, about the clinical data management, okay, drug development. So these things will be there. So just listen to those 4 hours classes in your free time, and also I'll be sending 2 or 3 PDFs to you. One PDF contains the role of the SAS programmer. Okay, what's the exact role of the SAS programmer and what type of terminology we use in the pharma industry. Then changing trends of the SAS programmer role, because like 7-8 years back the SAS programmer role was different, but nowadays the role has been changed and some new like works also allotted to the SAS programmer. So how, I mean, there's a, I mean, evolving the trends, okay, for a SAS programmer in the industry. So I have 2-3 presentation papers, the Suji papers, you know the Suji, right? So just go through the 1 or 2 Suji papers and also those 3-4 video classes. Then tomorrow directly I will start explaining you the programming part. Okay, so after programming part, and while I am explaining the programming part, then I will be discussing some theoretical concepts also. In that time you will be finishing those 4 hours class also, I mean 4 or 5 hours classes. Then we will start the SDTM ADAM, okay, and 2 hours continuously will spend on the statistics like that. Okay. So anyway, I will just send you the, you know, how we proceed also. Now I'm creating one folder and I will send you that folder link to your mail ID. So I mean, if you cannot receive that folder in 1 hour, okay, just get back to me, because sometimes it may go to the spam also. Okay, check the spam. Okay, so tomorrow we'll be starting the clinical part. Okay, and whatever the prerequisites I'm having, I will send you the material also, some books related to the, you know, the clinical trials, okay, some programs. So whatever you need, I just upload to the folder and I will send it to you. Okay, and
And one more thing I wanted